Hello, good afternoon. Myself, Dr. Nengraj S. Belagal. I am working as a assistant professor of entomology at the School of Agriculture, SR University, Anand Sagar, Warangal, Telangana State. Dear students, today we are going to discuss a very important topic. That's the insect endocrine system. Very important topic. Nowadays, it's one of the most important, as you know, most of the living things on this earth, they run one of the most important mechanism, or we can call that the hormones. Okay, these are very important micromolecules which are always present in the living system, and they will do some specific functions as they are programmed in our living system. So let's start begins with this lecture insect endocrine system. Okay, now we'll discuss what is the exact meaning of the endocrine system. Okay, great. Yes. Here, endocrine system. What the exact technical meaning of this? This endocrine is the word derived from the Greek word, where there are two words are there. First one, endo, and another one word is there, cranis. So what is endo? Inside we all know. And what is the cranis? To secrete. So it is a type of system which is present within our body or within the insect body, where the system of glands without any duct, where there are no ducts. Ducts are totally absent in the system, where the cells which are present in these glands, they are going to secrete some chemicals which are in the macromolecules. Okay, so that's why it's a system of glands without any duct, each of which secrete different types of hormones directly into the where circulatory system. That is nothing but our blood hemolymph or insect hemolymph to regulate for what? To regulate the various functions of the body. Yeah, this is one of the most important. Okay. So what is endocrine system? It is a type of system which is present in the living system without any duct. There are no specialized ducts are there to secrete or to burst themselves. But instead of that ducts, what happens here? Group of cells will be there. Okay. And those cells release some types of different chemicals, micromolecules, we call such as a hormones. Very important term, hormones. And when these chemicals or hormones they release from the specialized glands, those hormones circulate within our circulatory system and they go and they target or they will get attached to the specific receptors, and the organ which received that specific hormone will make it or it performs a specific function, and then the body system will run properly. So this is the total endocrine system. Next we'll deal with, I told you, these endocrine system they will release some macromolecules, and I told them as a hormones. Look at here, hormones. So what is the meaning of this hormone? Very important. Just to take 70 years before or 90 years before, we didn't know the meaning of the hormone. Okay, what we believed in the living system that something internal pressure is there that leads to functioning of some different organs functioning, and all the mechanism of living system will taking place. But there is a historical backgrounds are there in the next slides. I will give the lecture on that, how the hormones invented and what are the experiment conducted to exactly identify what are the hormones. So everything will be there. So now we'll study what is the meaning of the hormone. Okay, what it is hormone? It is the Greek word. Hormone nothing but I excite, I'll do something. Okay, so that is the meaning of the hormone. So if we define it, what is the hormone? It can be defined as chemicals produced in a gland, and that released into the blood system or circulatory system, and they have their effect somewhere else in the animal body. Okay, you look at this image. This is the endocrine gland. You just imagine, okay, and this gland releases the hormone. Then when the hormone release, where it release? It release in the bloodstream. Those hormones they degrade within the bloodstream. They move through the bloodstream and they go and target to the specific organs. Okay, and these organs after receiving a hormone, they will start functioning there. Whatever those tissues or those organs meant for the function, example kidney. Okay, if hormone released by our brain, so that hormone will circulate inside the blood system, go into the kidneys, nephrons, and those start functioning the secretion of the urine. So like that, this is the total glandular system. Glandular system nothing but endocrine system nothing but it's total related to the hormones, which these are hormones present in the inside body system, and those are we studying right now here. Now we know the what are the hormones. What are the some important functions of the hormones we will study. Hormones may result in embryogenesis. Okay, hormones, post-embryonic development behaviors. Hormones definitely they play in the water balance in the inside body or living system. They take place in the metabolism. They take place in the caste determination. Example, you take in the termite, honeybees. They take place in the polymorphism, different forms within the insect. Okay, they also help in the mating system according to their biological cycles, and reproduction damage, diapause they also take place. So all these, whatever the functions are there, okay, without hormones, these are unable to do. It's perfect. Hormones are the ultimate, where these organs they perform their activities when these specific hormones releases, and those hormones we are discussing right now. Next, see here, all the hormones, different types are listed in this one single image. So let's we study types of hormone release in insects. So I quoted with the individual, what you call A, B, C, D like that. So let's we study one by one. First you take, what's the A? A, here what happens? An endocrine cell releasing a hormone into the circulatory system. Okay, you just remember this is the endocrine cell. Okay, you just observing small dots, those are the hormones. Okay, they are releasing into the bloodstream. Any gland, just you take. Second one, B. A, where it is B, what it indicates? It's a hypothetical example of endocrine system. What are the different types are there? Okay, in B, a neuron synapse, it's there. Two neurons are there, 1 and 2. There is a small gap is there. This is called neuron synapse with neurosecretory cells. At the edge of the neurons end, okay, neurosecretory cells will be there. Release neurotransmitters within the synapse. You can also call this is the endocrine system. Okay, next you come to the C. Okay, neurotransmitters. Okay, a neuron synapsing with the endocrine cell releasing a neurotransmitter. You can also call it as the endocrine system. Like that, you just observe this image. Okay, where some specific glands they are releasing some micromolecules and they are hitting on target cells. So that's this target cell perform its biological functions. So that is the endocrine system. So now we'll study how the hormones get popularized. Before that, we never thought that there are the hormones within our body, they perform the functions. But only we are dependent on some internal pressure and they are doing such things and the body is performing the biological function. But very, very important. As I listed here, Battalion is a great scientist, physiologist in the 1894. He given the first evidence of insect hormones. Definitely there are the hormones are there. They are existed in the body of the living beings. So they play a great role in the performing the different functions by the different cellular structures or tissues. Okay. So what the battalion did, he just took, he just took silkworm larvae, that is Bombyx mori. Okay. He took the silkworm larvae Bombyx mori, and what he did, just observe here, this is the caterpillar. He ligated. He ligated at the just behind the head, or you can take just behind the thorax. Okay. So pupa is separated and head and thorax ends, the both are separated. So after 4 days, what he observed, only this part, head and thorax, they were pupated. Nothing but they went to the next stage of the 4 different stages of the caterpillar. As you know, this Bombyx mori is lepidopteran insect. They are having 4 different stages: egg, larva, pupa, and adult. Okay. When he ligated just behind the thorax, the abdomen not at all pupated, only the head and thorax they become blackened color. They are pupated. Okay. By looking at this evidence, what he thought that battalion, definitely there are some messengers within the insect body. They are going to do like such things. They are going to do make the insect to pupate after some critical period. He quoted to the external scientific authorities, but no one believed the battalion concept. Okay, first evidence of insect hormones. Next, the scientist we call the Kopek. During the 1917, he repeated the battalion experiment. What he done? Once again, just look at here. Okay, he took the Bombyx mori silkworm larvae. Okay, he ligated early during instar. He not the Bombyx mori larvae early, and he ligated late during the instar. Okay, when he tied the insect larva Bombyx mori early period, the head and thorax only they pupated, but not the posterior. Example, take 5 to 10 days within. Okay, he ligated. Only the anterior portion pupated, posterior is not pupated. And took another insect after 15 days. Okay, after 15 days, he ligated another insect. Here in this case, both head, thorax, and abdomen totally pupated. Head, thorax, and abdomen were total complete insect. The complete insect or larvae pupated. And he worried, and he not worried, means he got some surprise in him. If I do the ligation early in the larval instar, only the half portion is pupated. But the same thing when I ligated the insect at the later stage or at the fifth instar larvae, ligated, this larval stage converted into pupal stage successfully. So what he come with the results that definitely in the head region, there is a chemical is going to releasing. Okay, and when I tied this larvae, already that chemical released inside the insect body system, so that the macromolecules of the hormones reached the specific targeting receptor molecules, so that the insect completely larva become pupate. But that was not observed in the insect in the earlier stage ligations. So what he concluded from this experiment that definitely there are some chemical messengers they are present within the body, especially they are released by the head system. And with a specific time period, they circulate within the insect body that results into the pupation or different morphological characteristics. Example, taking the pupation. Definitely, there is a chemical, there is a hormone. Okay. Example, take molting hormone, but he won't know what is the molting hormone during that time. Now, present, I'm telling molting hormone after some time period, it circulates inside the bloodstream, released by the head, and this insect successfully pupation takes place. So from this experiment, what it decided, definitely there are chemicals are there, they called as the hormones. Okay, with time specific they release and results into a specific target oriented actions and the larval pupation takes place. So this was the two experiments. These were the two experiments. They found that hormones are there within the insect body, and that they results into the some specific actions. Those are called hormones. Next, at the final stage, Wigglesworth we call the father of insect physiology. Father of insect physiology. He done another one great important experiment we call head decapitation experiment. Head decapitation experiment. Okay. What he done, he took bug, that is predatory bug. That's bug name is Rhodnius prolongus. Okay. This is the can call also predator bug or parasitic bug. It will suck the blood from the animals, okay, and it will consume the blood. And that blood requires for its proper growth and development or physiological action of its body. So what he done, he took the Rhodnius prolongus bug, especially in the fourth instar nymph. Okay. So what he done, before 4 days of this fourth nymphal instar, he removed the head. So this is the head. Okay. He removed the head of the Rhodnius prolongus and the age of this bug during that time less than 4 days. Okay. So during this experiment, what done? This bug needs to go from fourth instar larval to fifth instar nymph. It must go. But in this case, this bug not transferred from fourth to fifth. It will remain the fourth instar larva itself only. And it took another bug. Okay, the fourth instar larva only. But he decapitated. He removed the head of this bug after fifth day, after completion of 5 days. He removed the head of the bug. So when he removed the bug head after 5 days, this fourth instar bug successfully become fifth instar name. Okay, it's converted fourth instar to fifth instar name. So from this experiment, what he concluded that there are some chemicals now hormones they release definitely from the head region. That's brain. Brain is the main organ in the insect, play a central dogma where it can releases or it cannot be releases and it take the control of over the complete body mechanism. So from this experiment, when you remove head, Rhodnius prolongus before 4 days, fourth instar larva not converted into fifth instar larva, it will be remained in the fourth only. And in the fifth, once again, when he took the fourth instar larva, but he removed the head of the bug after 5 days, but fourth instar larva become fifth instar larva. So from this experiment, what he concluded that during a specific time, hormone is released already, and those hormones circulated inside the body stream. And this, even though the head is not present, the body becomes fifth instar larva, fifth instar nymph. So from this experiment, he concluded that hormones release, mainly the releasing, not releasing, takes place from the brain itself only. The brain is the modulator for the complete set of the release of the hormones. It triggers in a particular time, and that results into the successful development of the insects. So this is the results for decapitation experiment. Next, now we will study what are the different types of endocrine systems they are present in the insect body. Okay, the different insect endocrine system. There are more than 15 endocrine systems they are present within the insect body, but among those 15, we will take very important 5 major endocrine system. If these are present, everything will be insect go on successfully life. Okay, so first one, neurosecretory cells in the brain system. So here, neurosecretory cells, you can also call the endocrine system part, because secrete, these cells secretes messengers or hormones also. Okay, example you take prothoracicotropic hormone, which is released by brain itself only. And this hormone triggers different glands to release from there itself and do some functions. Okay, so that's why among all the endocrine systems, the neurosecretory cells plays very important role. We can call mother of hormone site. Okay, next endocrine system is corpora cardiaca. What are these corpora cardiaca? These may be the group of two glands or single gland which is present just below the brain. So those are the corpora cardiaca. Next, corpora alata. These are the paired bodies which situated just below the corpora cardiaca. And prothoracic glands which are present in the thoracic segments. And next, the ring gland which is present just below the brain. So individual we will discuss one by one. That's very important. So whatever just I listed behind, so this is the graphical representation of all those endocrine system. Just to look here, this is the brain total. Okay, so the brain consisting of 4 different glands. Okay, so the outer one and the inner one, the inner one and the outer one. In this combination, they will take place, and those we call nervi corpora cardiaca 1 and nervi corpora cardiaca 2. So nervi corpora cardiaca 1 is from lateral and one is from middle, and this one is from lateral. Sorry, lateral plus middle. This one is lateral and this one is middle. They make nervi corpora cardiaca 2, and once again from middle and the last lateral they make nervi corpora cardiaca 1. So NCC1, NCC2, these are the cells we call the neurosecretory cells which are present in the brain in the 2 sides, lateral and middle. So these are the cells which are going to release the brain hormone. That's we call prothoracicotropic hormone. Prothoracicotropic hormone, very important. That's called the brain hormone, where the brain releases this prothoracicotropic hormone. And this prothoracic hormone immediately after releasing that's stored in a glands, and that's we call corpora cardiaca or corpus cardiacum. So what is the function of this corpus cardiacum? It stores it stores the secretions done from the brain. Okay, act as a storage and receiver organ within the endocrine system of the insect. So just below the corpus cardiaca, another pair of glands are there. They are the called corpus allatum or corpora allatum. Corpora allatum. These are the paired gland. Here, this side one and this side one. Okay. What they will do when the brain releases prothoracic hormone, this hormone triggers the corpora allata to release juvenile hormone. So, what is the function of this juvenile hormone? When there is juvenile hormone more inside the insect body, the insect will be in the younger stage. Sorry. Wait. Yes. The amount of juvenile hormone is greater extent in the insect body, the insect will be in the immature stages. That is nothing but the larval stages. And this juvenile hormone is released by the corpora allata, and that corpora allata release the juvenile hormone by the trigger of the brain itself only. Now, when this juvenile hormone little bit decreased a little bit, the early instar larva become later instar larva. Suppose you take, if it is the first instar larvae, that becomes second instar larvae, third instar larvae, and fourth instar larvae. Okay, means when the juvenile hormone goes on decreasing inside the insect body, the insect, immature insects, goes on maturing higher level. And when the juvenile hormone is very low inside the insect body, the larva get converted into pupa. Okay, it is the resting stage of insect body. And when the juvenile hormone is completely absent, now this pupa get converted into adult. So this is the importance of the juvenile hormone inside the insect body. It maintains the juvenility of the insect, youngerity of the insect. So that's why the larval stages or the nymph stages, they maintain a specific durations. Example, you take up to 28 days, up to 10 days. That days is mainly because of the presence of juvenile hormone inside the insect body. Okay, next. Now we'll deal with the neurosecretory cells in the brain. Okay. So as I told, they are the four in pairs, two in pairs. You just look at here. NCC1 median, NCC2 lateral. Okay. One median and one lateral, they forms together. Okay. And they continue downside, and another one median and another one lateral. They both together forms and they continue together. And this is the corpora cardiaca. And below this, pair of organs are there. And this is the corpora allata. Okay. So this is the structure. So these neurons are mainly made up of the motor neurons, neurosecretory cells in the brain. Nothing but the NCC1 and NCC2. They are mainly made up of the motor neurons within the ganglia, and they forms the glandular cells. These glandular cells, they release the prothoracic hormones. From what? From NCC1 median and NCC lateral one side, NCC1 median and NCC lateral other side. Okay, and these release the prothoracic hormone inside the brain cells. And this prothoracic hormone triggers the prothoracic gland. Just previous slide triggers the prothoracic gland. That's the hormone that triggers, that's called ecdysone, molting hormone. Okay, as I told you, first instar larva becomes second instar larva. How it become? Because of the only molting. If this ecdysone is present, then the first instar larva only becomes the second instar larva. Otherwise, The insect will remain in the first instar larva itself only. See how the functions of the brains are there. These two NCC1 and NCC2 releases the PTTH, prothoracic hormone, that triggers the prothoracic gland. These glands release their own hormone that's called the ecdysone hormone, and this molting hormone takes place in the molting of the immature stage of the insect. So if the insect want to grow from first instar to second instar, so this ecdysone play a greater role. Now, this first instar larva after 1 or 2 days becomes second instar larva. During that time, this prothoracic gland releases the ecdysone. Now it becomes first, second, third, and third to fourth. Simultaneously, juvenile hormone goes on decreasing, and during that time, where juvenile hormone is very less, but ecdysone is little bit more. I get pupation. Larva get pupation. Okay. After pupation, it emerges as adult. During this time, juvenile hormone is totally, completely absent. And ecdysone, very little bit it is present. Okay. Now, second, prothoracic glands. See, one of the important glandular system in the insect body, prothoracic glands. As I told, the brain is the ultimate modulator in this insect body, where it can release or it can trigger the release of specific hormones by specific glands within the insect body. So prothoracic glands, okay, they are the paired glands present ventrolateral part of the thorax. As you know, head is there, thorax is there, and abdomen is there. Okay. Ventral side, just your chest, below the side, and lateral side. Below the insect, at the side of the insect, they are always present in the prothorax only. Prothorax means the first part of the thorax. Three types of thorax, three segments are there in the thorax: prothorax, mesothorax, and metathorax. Okay, in the prothorax, they are situated, first part of the thorax. They are also called pericardial or ecdysial glands. Why we call ecdysial glands? They take place in the ecdysis. We know what is ecdysis. Setting up the old skin. That's we call exuvia. You might also observe in the snakes. They will call it outer skin. Exuvia we call that. Okay. So this prothoracic gland, they are not present in the adults. Only they are present in the immature stage, because one stage of the instar get converted to second stage, second to third, third to fourth, up to seven stages in some insects. If they won't take conversions, definitely insect won't grow and they will die. So not to do that continuity, okay, this prothoracic gland plays an important role. This prothoracic gland releases the ecdysone, molting hormone, by the trigger of the brain, what you call prothoracic hormone. That hormone triggers the prothoracic glands to release molting hormone ecdysone. When the ecdysone release, insect goes on molting. Here you see molting hormone, they secrete prothoracic gland. We call that ecdysone. Next. Here, some of the prothoracic glands listed here. In the case A, present in the cockroach, they are scissor-like present in this side and that side, and intermittently they are crosswind. But in the hemiptera, they are present in both lateral side, this one on this side. And in the lepidoptera, in the thoracic regions, they are present one side pair and other side. And in the hemiptera, they look like tree. Okay. So these are some images where the prothoracic glands they are present. In Lepidoptera C, they are highly developed, because especially the prothoracic glands are highly developed in the holometabolic insects, where the complete set of all the four different stages are there present: egg, larva, pupa, and adult. Because in the holometabolic insects, the larval stages are greater in extent, they need to preserve the food molecules a lot. So there are more instars will be there. So to carry successful those instars, to get successful life, the molting gland play a greater role, and they release the ecdysone, and that ecdysone triggers molting process, and insect completely succeeds juvenile stage or young stage. Here, given one term agrees to rights. This is the generic name, chemical name given to the hormone ecdysone, where the artificially synthesized, prepared. So such term we call active steroids. First time Buchanan and Carlson in 1954 extracted such type of chemical molecule. We call that steroids or steroid hormone, which is in the crystalline form from the Bombyx mori. Very, very important from the exam point of view, students. The artificial or the extraction of this hormone, ecdysone hormone, done by the Carlson and Buchanan in 1954 from the external source. Okay. So what this ecdysone will do, they act against on them itself only. The more the ecdysone hormone, okay, insect will die because it causes a prolonged molting period. Insect never takes the pupation period. So if there is obstruction in their proper four different life stages, insect will go, they will die. And extraction of those molecules artificially, and if you spray on the crops when the insects are present, so that they never get pupated, they never get they spread their population. So the insect will die. See how these are all the some environmental friendly. You can use such things in the integrated pest management. That's why these steroids develop. Most safest to the environment. Nothing happens. They are the species specific. They only act on such insects and they will get killed. No harm to the any animals, birds, or it may be the human populations. So very good one. So in the what you call steroid hormones, two types of there, especially in ecdysone, alpha ecdysone and beta ecdysone. Okay, where high dose of beta ecdysone cause sudden death of the insects within 3 to 4 days because they disrupt the life cycle and they will get die. Now we'll move to the next important endocrine system organ, corpora cardiaca. This structure present just below the brain. Just to observe, it's the brain. It's the brain. Just below the brain, the corpora cardiac is present. It is either in the paired or in the single. Okay, not like structure is there. And this is associated with the heart, aorta, and situated immediately behind the brain. It's absent in the columbola. Okay, please remember. So, what is the function of corpora cardiaca? As I told you earlier, whatever the hormones released by the brains, NCC1 and NCC2, there is the PTTH, prothoracicotropic hormone. It's the brain. Okay, this hormone released in the brain. This hormone get and collected inside the corpora cardiaca. This corpora cardiac also produces and releases its own hormone, such as adipokinetic hormone. Okay, adipokinetic hormone, its own hormone from the corpora cardiaca. And another one of the important most function of the corpora cardiac is it controls heartbeat in the insects. Very, very important. It controls heartbeat in the insect and it regulate the glucose level in the insects. We call the tree halose. The sugar of the insect blood is tree halose. It controls the sugar content of the insect. Very, very important. Not only release the adipokinetic hormone, it also releases some different hormones also, and along with that, it controls the heartbeat as well as the insect blood sugar level. So that's the corpora cardiaca. Now we'll study corpora allota. Corpora allota. This is the organ just present below the corpora cardiaca, brain, corpora cardiaca, next below corpora allota. These are the paired organs. They are always in the paired organs. And these organs first discovered by the Janet in 1899. They are usually ectodermal in origin and they are glandular bodies. They secrete their own molecules. In hemimetabolous insects, hemimetabolous insects, where the partial metabolism is there, contains large number of small glandular cells, but large and few in number in the holometabolous insects. there is little bit modification is there. So what is the main important function of the carporolata? They release the juvenile hormone. Very, very important, as I explained earlier. It what the function of juvenile hormone maintains, it maintains juvenility. Nothing but larval structure. Maintains the young stage of the insect itself only. After egg, larva hatches. So larval period, it continues from first instar to fifth or seventh instar. That maintenance of the larval period is mainly because of the presence of juvenile hormone. It also regulates the metamorphosis. Nothing but change in the form. First instar form to second instar form, second to third. So this is the main important function of Carpora alata. And another one of the most important function of this is yolk synthesis. In the female, egg production takes place. Okay, every egg is having yolk. That is the protein material. Protein, yellow color in there. Protein material. That yolk synthesis is mainly done by the Carpora alata itself only, and that is the juvenile hormone. These hormones stores the yolk material inside the egg that act as a reservoir for the next generating any insects, and reproduction in the oocytes of the adults takes place. Look at here, these are the corpora alata. Two paid organs and one single. They may also have one single organs. They are present in the hemiptera as a single modification, but in most lepidoptera, they are in the paid. Another one important, endocrine glands. That's we call ring glands. This is the very important gland where what happens up to now, where the corpora cardiaca, corpora alata, and prothorax, what you call nervi corpora cardiaca 1, they are all individual separate. They are arranged. We call the name ring gland. What happens here? All these organs, they were explained. Corpora cardiacum, corpora alata, and prothoracic gland, all they are arranged in a circular fashion. They are clubbed within a circular ring-like structures. That's why we call ring glands. And usually these glands are present in cycloprus insects. And these ring glands connected by some specialized nerves, they connected to the brain. Okay, they connected to the brain. And when the brain triggers PTTH, this ring gland takes place the proper functioning of all the maintaining of the juvenility, NIM stages, okay, and sometimes pupal stages also. So this is the specialized gland where all the organs, corpora cardiaca, corpora alata, nervi corpora cardiaca 1 and 2, they club together, they form in the circulars. So that is the ring gland. Another one is there epitracheal glands. Epitracheal glands. These glands are usually they found in the lepidopteran insect, especially in the caterpillar stage itself only. So where example take Munduca 60, okay, where they are located, they are present surrounding the spiracles. These spiracles. Usually the complete 8 spiracles are there in the abdomen segments. They are present surrounding the spiracular areas. Epitracheal glands. So what is the function of these epitracheal glands? They act as a source of ecdysis triggering hormone. Ecdysis triggering hormone or occlusion in Lepidoptera. We know that what is ecdysis triggering hormone. So insect usually grows by molting. Okay. So when insect want to molt, it needs to rupture its outer skin and need to remove that outer skin, and fresh skin it takes place and insect grow. So when the old integument want to release from its body, so this is the hormone, ecdysial epitracheal glands. They release ecdysis triggering hormone. This is the ecdysis triggering hormone. After releasing from the ecdysial epitracheal glands, the ecdysis triggering hormone released inside the blood system of the insect. And this ecdysis triggering hormone comes and deposits at this epitracheal gland so that the rupturing of the skin of the larva takes place from these glands. So the old skin is removed and the old new caterpillar comes outside from this insect. So that is the important function of epitracheal glands. These glands releases ecdysis triggering hormone. So the larva molt, old skin removed, the ones become a newer one. So these are the importance of epitracheal glands. So now we come towards the last part of our topic. Okay, that's the summary part of the topic. As you study now, endocrine glands. As you know, these are the glands which are present within the living system. There is no duct, ductless glands. Okay, ductless glands. So when there is no duct, what happens? These glands having some tissues, okay, and they are having the cells, and these cells releases hormones. Example, you take the brain having brain hormone. So these brain hormones released by four different types of cells, 1, 2, 3, and 4. Okay, you call nervi corpora cardiaca 1 and nervi corpora cardiaca 2, where one from this and one from this they make and they go downside. Okay, and one from central and another from lateral and they go down here. Okay, so these four different cells we call nervi corpora cardiaca cells. They release the brain hormone and that's called prothoracicotropic hormone. Okay, what the main function of this prothoracicotropic hormone? As you know, this hormone is released by the insect brain system by the four different types of cells to lateral to inner. So this prothoracicotropic hormone triggers a release of the molting hormone. Okay, release of the molting hormone we call that ecdysone from what? Via prothoracic glands. These are the prothoracic glands. So this prothoracic tropic hormone act on the prothoracic gland. This prothoracic gland releases the molting hormone that's we call ecdysone, and this ecdysone act on the insect larva of caterpillar so that the first instar goes to second instar, second to third, and third to fourth. Molting we call molting, removing of the old skin and formation of new insect larva. This is the main function of the prothoracic glands, and the release of this ecdysone mainly done because of the trigger of the prothoracic tropic hormone. Now it's one side. Okay. Now, once again, this prothoracic tropic hormone act on the corpora allata. Okay, and these are the corpora cardiaca. Where corpora cardiaca they store brain hormone. Okay, and also they do its function heartbeat control, adipokinetic hormone release also. Okay, now it's a prothoracic tropic hormone act on the corpora allata, and this corpora allata releases the juvenile hormone. When the corpora allata releases juvenile hormone in greater amount, okay, then the insect is always in the young stage. That's we call larvae. When the juvenile hormone goes on decreasing, now the insect goes on adulting or aging. So first insect to second, second to third, third to fourth. Okay. When the juvenile hormone is absolutely very low and insect now become pupae. And when the juvenile hormone is totally absent, now the insect become adult. So that is the function of corpora allata. This one of the endocrine system releases the juvenile hormone, and the insect molting along with the juvenility takes place with a different morphological or life cycles takes place. So this is the in summary. Brain is the central dogma, releases the prothoracic tropic hormone that act on the prothoracic gland. This prothoracic gland releases the ecdysone, molting hormone, insect molts. Once again, brain releases the prothoracic tropic hormone. It triggers the corpora alata. Corpora alata releases the juvenile hormone. Insect molting takes place. First instead of second, third, and fourth. When the juvenile hormone is high, the juvenility will be more. Young instars will be the more caterpillars. When the juvenile hormone is less or very low, pupation takes place. When absent, adult takes place. So likewise, the insects, they take successful their different life stages and they will get succeed on this earth. So with this, I will stop here. Still a lot of, this is a very big chapter. If you take only one hormone brain, so it can deal with the 5-6 hours. Now we'll stop here. And thank you for listening this lecture. Thank you so much. Thank you.